Assalamualaikum warahmatullahi wabarakatuh. Good morning everyone. I would like to tell you about myself. My name is Saumi Kurnia. My hobby is traveling and I live in Jakarta. How is your life? And now I want to explain material about photosynthesis. And happy watching and enjoy this video.
Material about photosynthesis. First, definition of photosynthesis. Second, the process of photosynthesis. Third, reaction of photosynthesis. Fourth, determinants of the rate of photosynthesis, and the last, effect of light on chlorophyll. Definition of photosynthesis. First, photosynthesis is a chemical reaction that takes place inside a plant producing food for the plant to survive. Second, carbon dioxide, water, and light are all needed for photosynthesis to take place, and the last photosynthesis happens in the leaf of a plant. History of photosynthesis. The history of studies done on photosynthesis dates back into the 17th century with Jean-Baptiste van Helmont. He rejected the ancient idea that plants take most of their biomass from the soil. For the proof, he performed willow tree experiment. He started with a willow tree of 2,27 kilograms. Over 5 years, it grew 16,7 kilograms. However, the weight of the soil only decreased by 57 grams. Van Helmont came to the conclusion that plants must take most of their weight from water. He did not know about gases. Joseph Priestley ran a series of experiments in 1772. He tested a mouse, a candle, and a sprig of mint under hermetical seal. No air can go in or out. He first observed that a mouse and a candle behave very similarly when covered, in that they both expand the air. However, when a plant is placed with either the candle or mouse, the plant refreshes the air for both. Further ideas were brought about in the late 1700s. Jan Ingenhousz and Jean Genever found that the air is only living in the daytime and that CO2 is assembled by plants. Antoine Laurent Lever found that rarefied air is a separate gas, oxygen. The process of photosynthesis. First, plants need food to respirate, grow, and reproduce. Unlike animals, plants are able to make their own food by the process of photosynthesis. Second, photosynthesis takes place in the part of the plant cell containing chloroplast. These are small structures that contain chlorophyll, and the last, for photosynthesis to take place, plants need to take in carbon dioxide from the air, water from the ground, and light, usually from the sun. Photosynthesis reactions are generally divided into two, namely, first, light reaction. Light reaction is photosynthesis that occurs in the chloroplast liquid membrane. The light reaction, the first to claim the role of light in photosynthesis, was Van Hill, an American biologist. He researched photosynthesis in purple sulfur bacteria and came to this conclusion. These microorganisms produce gases from CO2 as well as green plants. And to obtain these microorganisms need light. However, water is not used as a starting material, but these bacteria use hydrogen sulfate or H2S. In addition, during this photosynthesis, no oxygen is released, but elemental sulfur is found near argued that the action of glycos that the composition of H2 into hydrogen and sulfur atoms. Then in the dark reaction, hydrogen atoms are used to reduce CO2 in a series of dark reactions. He argued that in current plants, like energy gases water to break down into hydrogen and oxygen. Then the then the hydrogen atom is used to reduce CO2 in a series of dark reactions. If this theory is correct, the conclusion would be that the oxygen produced in photosynthesis comes from water just as all sulfur obtained in the process of bacterial photosynthesis is derived from H2S. Second, the dark reaction. The dark reaction is a reaction in photosynthesis that takes place in the stroma and does not require sunlight as this energy source. The dark reaction in photosynthesis is actually a series of reactions that involve uptake of CO2 by plants and reduction of CO2 by hydrogen atoms. Dr. Kelvin and colleagues at the University of California investigate for years the step-by-step sequence of the chemical reaction in fluid. The basic experimental product is to expose a suspension of unicellular green algae to light and radioactive carbon dioxide. The use of radioactive carbon 14 on carbon dioxide throttles the atom, making it possible to study its chemical transformation to determine which substance, if any, of the separate substances on the chromatogram are radioactive and X-ray film is placed near the chromatogram. If black dots appear on the film because there is radiation emitted by 40 She atoms, their position can be correlated with the position of the chemical on the chromatogram. With this autoradiography technique, Calvin found that 40C appears in close molecules 30 and 30 seconds after the start of photosynthesis. If this was allowed for the photosynthesis to last only 5 seconds, he would find the radioactive in other smaller molecules. Gradually, the path of carbon fixation can be determined. One of the important substances in this process is the 5-carbon sugar, which is phosphorylate, namely ribose substrate. Determinants of the rate of photosynthesis. First, light intensity, the maximum photosynthesis rate when there is a lot of light. Second, carbon dioxide concentration. The more carbon dioxide in the air, the more material plants can use to carry out photosynthesis. Third, temperature enzymes that work in the process of photosynthesis can only work at their optimal temperature. Generally, the photosynthesis rate increases with increasing temperature to the limit of enzyme tolerance. Fourth, moisture leach of water or drainage causes the stomata to close, inhibiting carbon dioxide absorption and thus reducing the rate of photosynthesis. And the last, photosynthesis level or photosynthesis result. The level of photosynthesis such as carbon dioxide are reduced, the rate of photosynthesis will increase. Effect of light on chlorophyll. When a chlorophyll solution is added in a beam of light, it will give off a dark red light. This phenomenon is tinged with fluorescence. This can be easily demonstrated. The crude chlorophyll extract can easily be demonstrated. The crude chlorophyll extract can be prepared by dipping the grass leaf in ethanol. In a white streak of light, this solution shows fluorescence. The explanation for this phenomenon is that the absorbed light energy is transferred to an electron in the chlorophyll molecule, raising it to a higher energy level. These electrons are free to move around the molecule. These electrons are usually left to high energy level, so the chlorophyll molecule is called excited. In the chlorophyll solution, the aerosol stage passes quickly. The electron returns to their original energy levels. Thus, the electron gives off the energy that left it in the beginning. Most of this energy is released as heat like with very clear vessels.
Enough material today. If you found many mistakes, please forgive me. Let us say wassalamualaikum warahmatullahi wabarakatuh. Don't forget to like, comment, and share this video. Bye bye.